Well, thanks very much for the invitation to come to speak at this event. And what I would like to do this morning is talk to you a little bit about small grains. In particular, my position at the University of Minnesota has been working on barley breeding for the last 17, going on 18 years. So I'm going to focus mostly on barley and barley breeding, although recently I have taken responsibility for restarting our oat breeding program. So I'm going to talk a little bit about oats as well. And the title, Mac had given me a whole list of different things to talk about. And so I'm going to focus mostly on breeding and varieties, some disease resistance things, and not as much on agronomy, but I have a few things I can say about some of the differences that are in terms of growing, say, malting barley versus oats or wheat that I'll kind of sprinkle in here. But again, the focus will primarily be on breeding and genetics and varieties. I also put my email address up here, and I really do encourage folks who are interested in learning more about what's happening in the program or have a thought of ways that they could participate in collaborative research with the university. Myself and a lot of my colleagues are very much interested in forming those partnerships with folks who are actually out in the field growing these crops. And so we do have a number of on-farm collaborators, but we're always looking for new collaborators, both within the growing community, but also in the malting and brewing community as well. We look for those sorts of collaborations because really at the University of Minnesota, we're a land-grant university, and our mission is to do research that's relevant for all of you. So please don't hesitate to contact me if you've got questions or ideas. I would love to hear from you. And I think that goes for my colleagues as well. So you can easily, we're all very accessible. You can go to the University of Minnesota website and find most of us pretty easily. Let's see.
Yeah, the green circle just hit on the right side of it.
Aha, gotcha. Okay, so what I'm going to talk about this morning is a little bit about barley and oats. And in barley, I'm going to talk about 6-row spring, 2-row spring, and winter barley. So we have a couple of different breeding programs, and that's changed over the years. And I'll talk a little bit about the work that we're doing to restart the oat breeding program, which has been fairly quiet for the last 5 or 6 years. I also want to make sure that I, right up front, acknowledge a lot of my collaborators at the University of Minnesota. We have a really great group of small grains researchers at the university. And so I have the pleasure of working with a couple of different pathologists who can help us with a lot of the disease problems that we have in small grains. Joachim Wiersma is the main person who's really the agronomy expert. So for those agronomy questions, he's a good person to rely on. I work on with a couple of folks, Gary Muehlbauer and Peter Morell, on sort of more basic genomics and genetics. And one of the things that we're trying to do is really understand the barley genome, and eventually I think we'll be working into the oat genome to see what we can learn from how the genes are organized in these two crop plants, and use that to develop technologies that will help us to better develop new traits or improve the traits that we're currently working on. So this is just sort of a little timeline. The University of Minnesota has been involved in barley research for over 100 years, breeding research. And most of that history, the focus has been on spring 6-row malting barley. And so there's a list of, not all, but many of the varieties that were released over that time period, and that's back to the early 1900s. More recently, we have diversified our breeding effort to include not only 6-row spring barley, but also 2-row spring barley, and I'll talk a little bit about the difference between those two. And then also winter barley as well, both in 6-row and in 2-row. And very briefly, the 2-row interest has really been sparked by changes in the malting and brewing industry. The large brewers like Anheuser-Busch, Miller-Coors have typically used a mixture of 2-row and 6-row malting barleys to produce beer, But over the years, that trend has switched to using more 2-row and less 6-row. So the industry has been telling us that they have more of an interest in 2-row. It's not that 6-row is going away. I think it has a place, and certainly there are brewers that are very much using 6-row. The other thing is that there's a large craft brewing industry that's emerging, and they have mostly have a strong interest in 2-row barley. So we try to pay very close attention to what's happening out in the malting and brewing industry so that we can direct, our breeding efforts. And then the winter barley effort really stems from the the very recent interest, I think, in developing cover crops for Minnesota agriculture. And I think many of you probably are aware the governor has a strong interest in cover crops, buffer strips, water protection, things like that. And so there are a number of folks at the university that are really interested in expanding our research initiative to say, okay, what other kinds of crops can we put out there that are either perennials or winter annuals that could be part of what's grown on the landscape to help to deal with some of the water quality issues, wildlife habitat issues, improving soil health and things like that. There's a sort of a broad umbrella name for that initiative called Forever Green. So if you want to know more about it, you can just Google Forever Green at the University of Minnesota, and you'll see there's a number of different projects involved in looking at what are some new crops that could be out there as tools that farmers could use in their own systems. And I think if you look at the history of cover crops, and I know you're going to hear a lot about cover crops this afternoon, they have had a limited success primarily because there's not a huge economic incentive to use cover crops. And so one of the things that we're really trying to think more carefully about in the Forever Greens initiative is how do we create that economic value to cover crops so that they're profitable and they accomplish all of the other things that we're trying to do with cover crops, including keeping continual cover on the land throughout the entire year. Okay, so just kind of a little snapshot or a picture of what barley variety pictures look like. This is a chart that shows you the percent of the acreage that's been grown in different varieties. And it's not important that you know the different varieties up here, but one thing that I think you'll see quite clearly is that in general we have relatively few varieties. Very different from corn and soybeans where you have dozens of varieties to choose from each year, and those varieties turn over every year. Malting barley is very different. The malting brewing industry has very specific varieties that they want to use, and they basically treat their brewing methods as very careful ingredient lists that they use, and they don't like to change those ingredients very often. So once they find a variety that works quite well in the malting process and in their breweries, they kind of tend to stick with that variety. And that's what you see. You see varieties that stick around for quite a long time, and Robust is sort of the king, which lasted for 20 years as a very dominant malting variety in the Midwest. So there's not very many out there to choose from at any one particular time, and they tend to stick around for quite a long time. I mentioned this earlier, but this slide is outdated. It gets outdated almost every week. I think we have in the state of Minnesota now over 100 breweries in the state of Minnesota. And most of those have just sprouted up in the last couple of years. So this is just a little map. You can go to the website from the Growler magazine, and they keep updating this list. And you can see there's all kinds of breweries scattered around the state, particularly around the Twin Cities area. And this is a really interesting new development for the industry in terms of what I think about as a barley breeder, because this is a very different end user group from the large brewers, and their interests and what they're looking for in varieties is probably different. And they're a new group, so it's something that we have to discover, what they're interested in. But one of the things that they are very interested in is locally produced barley. You can talk to just about any small craft brewer, and if you give them the choice of sourcing their ingredients locally, that's a really important thing for most of them. So to me, I think that provides some of the opportunity and some of the drive to think about how do we take our breeding research and our agronomy research and think about how we can make barley more profitable and productive and serve sort of a local industry. So this is this is just statistics from the Brewers Association. So Brewers Association is a large group that represents all of these craft brewers. Like I think they have over 3,000 members nationally. And they put together a lot of stats on beer consumption and beer production. And I'm not going to go through all of this here, but you know, at the time that this was put together, they said there were 73 craft brewers in the state of Minnesota. And the amount of beer is they're making 528,000 barrels of beer per year, so that's what we're producing. So I just did a little back of the envelope calculations to say, okay, well, if we wanted to produce all of the barley needed for Minnesota's local craft brewers, what would that amount to? And so based on the amount of barrels and how much barley is needed to produce the malt for those barrels, I came up with something like you know, just under 20,000 acres is basically what it would take in the state of Minnesota to supply that effort. So it's not a huge acreage, but it's a fairly substantial acreage. And again, this is a growing industry, so that number will likely continue to grow. And there might be opportunities really for premiums for locally grown barley for some of this craft brewing industry. So something that I really want to keep an eye on and again, continue to work with the brewers to get them sort of connected with growers through the work that we're doing at the university. Okay, so I mentioned 2-row and 6-row already, and if you're not familiar with barley, this might be something that's not readily apparent. 2-row and 6-row barley are basically described because of the shape or size of the spike that's produced on the flower or flower. 6-row barley, I have a little schematic here. So those are the pictures of actual plants. My graduate student did sort of a cartoon picture of that, but 2-row barley, you basically have 2 rows of kernel extending up the spike, and the lateral flowers, which are depicted in the sort of open circles there, are sterile. They don't actually develop. Whereas in 6-row, you get this whole row of kernels all the way around the spike, and you get this sort of rounder, fuller appearance of the spike. And these really are treated as 2 separate commodities. Their breeding programs are usually separate, so you have a 2-row program and a 6-row program. Typically, as a breeder, you don't make a lot of crosses between 2-row and 6-row. I already mentioned that the malting brewing industry has very strong preferences for 2-row or 6-row. The larger, big brewers that are more adjunct brewers have tended to like 6-row malting barleys. A lot of the craft brewers tend to like 2-row. So they're really different animals in that sense. But what's really intriguing is when we look at the genetics of the difference in this flower structure, it's really due to just a single gene. So, you know, you can actually make a cross between a 2-row and 6-row and easily find, you know, either 2 rows or 6 rows in that cross because it's just one gene controlling that. But in fact, the breeding history of these 2 are so separate that there's quite a lot of differences between 2-row and 6-row. Okay, so just a little more sort of background in terms of what's going on in the industry and what we're thinking about in terms of barley. Barley production has moved much further north and west, so most of the barley that's produced in the state of Minnesota is up in the northwest corner, and it's really been driven by economics. Corn and soybeans are strong competitors with small grains, and so when those are more profitable, that's going to be what's grown, and that's really what dominates a good share of the state. But one of the challenges is that a lot of the large malt plants still reside in the Midwest. And so some of you may be aware of the company RAR Malting. It's in Shakopee, Minnesota. So RAR Malting right now, they just did a big expansion of their malt plant. They are, I think, the largest single-site malt company in the world right now in terms of their production. So they're a very large producer of malt. And when I talk to them, their big challenge is that they are sourcing barley from further and further away. They're railing it in from western North Dakota or even in from Montana. And as you can imagine, the transportation costs are quite expensive for them, so it adds a lot to the cost of their production. And they're very much interested in trying to source more barley locally. In fact, I can share one sort of little anecdotal story. I was working with an extension agent just across the river in Wisconsin near Alma, who had a few growers that were interested in trying to grow barley again. They hadn't grown it for many years, and they said, we'd like to grow some malting barley, and we know there's some brewers locally in Wisconsin that are interested, and so I got them connected with RAR. RAR set up some contracts with them, and the last 2 years they've grown barley. It's a small amount of barley, and had it malted at RAR, and it malted very well. The RAR was very happy with it. And Nuglaris Brewing in Wisconsin basically bought up all of the malt right away. So they're very interested in local production, locally sourcing, being able to say they have Wisconsin barley in their beer. So again, it's just an anecdote, but it tells you that there is some interest out there, and I think there's some opportunities. Not that there aren't challenges with growing barley and getting malting. Yeah.
Can you sort of connect the dots for those of us that don't really understand how the malting takes place? So do the independent brewers, these microbreweries, craft brewers, do they ever do their own malting, or do they always go to RAR for their malt?
Yeah, great question. So the question's about malting, sort of the malting and brewing process. In general, those are two totally separate industries. So you have malt companies and you have breweries. So RAR Brewing, or RAR Malting, for example, has a spin-off company that does source, they call it specialty, I can't remember the name of it, but they do source malt to craft brewers. So they're a big malt company, but they have a spin-off and they do source to small craft brewers. And then there's a malt company called Breeze in Wisconsin that specializes in specialty malts. So when you look at all the different kinds of beers that are out there produced right now, basically there's a base malt that's most of the malt that makes up the beer, and then they add a specialty malt to that, and it's usually a small amount, and you can think of it as sort of like dark roast coffee, right? They roast it at different temperatures and they get different flavor compounds out of it, and they'll add a little bit of that specialty malt in part of that brewing process. So the specialty malts they might buy from Breeze, or they might import from Europe, depending on that sort of thing. Yeah.
Is the malt sprouted first and then roasted?
Yeah, I think I have a slide coming up, and I can talk a little bit about the process. Yeah, absolutely. So I think I'll get to that in a second, but I just want to give you kind of a feel for how we run our breeding program and the kinds of locations that we're testing at. So these are the locations that we have sort of permanent sites tested at. I work with Joachim Wiersema, who's an agronomist who's stationed up in Crookston, a part of the University of Minnesota, and he's developed a network of some on-farm trials so we can test with that. And then I also work very closely with the breeder at North Dakota State University so we can test things across a broader region. So this is the way that we can get a good handle on how the experimental lines that we produce in our program perform across a wide range of environments, of growing environments. We have a lot of traits that we look at. I'm not going to go through all of these, but you can think about them as sort of your basic agronomic traits. Obviously, yield is very important. Because of the malting and brewing industry, we have a lot of malting quality traits, and this isn't really even all of them. These are sort of the ones that are the most important. And actually, my next slide is going to get to the process of malting and brewing. And then we have a number of diseases that are particularly important. And I'll mention two of them. Fusarium head blight and bacterial leaf streak are probably the two most important diseases. Fusarium head blight, or head scab, has been around for a while now. Early 1990s is when it became really a serious problem again. So there's been a lot of effort developed to manage that disease. And basically, the choices for management are try to pick a variety that's a little more resistant, and there are some varieties that have a little more resistance, although that's something that we're continuing to work on. Fungicide applications are somewhat effective, but not completely effective. And then the big thing has been a changing practice in terms of rotation. So not following corn is a big thing. So corn is a host for Fusarium, the pathogen, and it survives on the residue of corn. And so if you have a lot of corn residue on the ground and you plant small grains into that, you're basically setting yourself up for disease problems in the future. So that's the main ways to manage. It's heavily influenced by the environment, so rain events. And there are some prediction models that are out there that are very easy to use to say, okay, I planted this variety at this date, it's flowering at this time, what's the likelihood that, given the weather patterns, what's the likelihood that disease is going to occur in my field, and you can use that information to make decisions, sort of last minute decisions about applying a fungicide or not. Although I have to say most of the growers who are still growing both wheat and barley in Minnesota are planning on a fungicide application for this particular disease. Bacterial leaf streak is a really different disease. It's been sort of a more recent problem in the last 5 years. It's caused by a bacterium, so fungicides do nothing for it. So you cannot control this disease with a fungicide. And unfortunately, there are not very big differences in barley for resistance to bacterial leaf streak. In wheat, there are some differences, and there are some varieties that are a little bit more resistant. And the Minnesota Experiment Station publications with the varietal guidelines clearly indicate which lines are more resistant to bacterial leaf streak. So then to the malting and brewing process. So these are really two separate processes. And the malting process is sort of broken up into these 4 steps. You clean the barley, you steep it in these steep tanks. So this is an example of a steep tank here where the barley is basically in a big tub of water that's aerated. And then once you get enough water uptake into that barley, it's dropped down into these germination areas. So this is a warm, humid place where the barley is kind of rotated over and over, and you get germination occurring. And so exactly what you were describing earlier, the seed is germinated. You can see the little roots developing on this germinated malt. And then it goes to a kiln, and then the kiln raises it to a certain temperature, dries the malt down, and the little roots will fall off actually at that point. So when you get malt, it looks very much like seed that comes in. It looks very similar. It might have some color differences. And again, you can vary all of these steps to sort of change the characteristics of that malt. And different breweries will have different requirements for the kind of malt that they want. So they'll say, we want this variety, we want these characteristics, and then the maltster will figure out how to malt it to meet those specifications. And then the brewing process involves milling that malt, making a mash. So these are actually my graduate students at my house doing a little home brewing, something I just learned how to do this year actually. And actually they were teaching me how to do brewing instead of me teaching them. I teach them genetics, but anyway, they're pouring out a mash right now we just finished boiling that, and we're pouring it into a fermentation. But so then you mill it, you mash it, you boil it, you add hops, you chill it down, you add yeast, and then it goes through a fermentation, and that's that whole brewing process. So it's a really complicated process where lots of things can go on, and so that's really the reason why brewers don't like to switch their varieties very often, because this is, they sort of tailor their process to those varieties. Okay. I won't go through the details here, but just so that you know, we talk very closely to the American Malting Barley Association, which is an association of, used to be really large brewers, but now there are actually some sort of medium-sized and craft brewers in there as well as the Brewers Association, and they have set up guidelines for us. So when we develop new varieties, we know what they're looking for in terms of the barley protein level, the level of beta-glucans, enzyme levels, the amount of free amino nitrogen. So there's a lot of different parameters that they measure that are important for them, and they have specific guidelines for whether it's 2-row or 6-row. As an adjunct brewer, so an adjunct brewer means that they use malt, but they also add other things like corn or rice or corn syrup or grits to that process. And so Anheuser-Busch is an example of a large adjunct brewer, or all malt brewers, and that tends to be where the craft brewers are. They use just malt. They don't add any adjuncts. And so they have a little bit different profile of characteristics that they're looking for. And those are the targets then for our breeding efforts. Okay, I'll go through a little bit of our breeding pipeline, and I'm just going to go through it for barley, but it's quite similar for wheat or oats too. There's a lot of similarities. But it all starts with making crosses. So here's just a picture of, this is a barley floret. So we actually snip off the top of it, and then this is the inside of the flower. These are the anthers, and we remove the anthers, or the male portion of the flower, leave the female portion there, and then we'll come back a couple of days later, take anthers from a different plant, and then we'll pollinate that flower. So it's a fairly tedious process. I have a couple of technicians who are really good at it. My eyesight's getting really poor, so I don't do very much of this anymore, but that's where it all starts, is picking parents and actually selectively crossing between the two. And then we have this sort of a pipeline here where we make crosses, we self-pollinate for a couple of generations, we evaluate the different lines that we develop, and we select for a number of different things, and then we go into an industry testing program where the industry can actually take small samples of barley, malt them, look at the characteristics and then decide, and get to a release. And that's like about an 8-year process. So it's not a fast thing. That's why it's really important to sort of know where your targets are, because you want to know where your targets are in 8 years from now. So hopefully they're not changing too much. So we often describe that as kind of a pipeline. I also like to describe it as a funnel. And so the reason it's a funnel is that at the early part of this pipeline, you're looking at really many, many, many different progeny from lots of different crosses, maybe thousands of progenies. And the process of breeding is just to sort of select among those and slowly winnow those down to where you're really just looking at 1 or 2 that you're going to consider releasing in any particular year. So that's kind of how that works. And then I'll just show you, well, one of the things that's a consequence of that is that you have a lot of genetic diversity, or you have a lot of different characteristics that you can select for here, but when you get down here, it's fairly narrow. So there are different things that we can select for at different stages of the pipeline, and I've kind of indicated them here. I'll just kind of show you a few things, but the selection is really based on a couple of things, sort of time and money. When you have 10,000 things to select among, you have to make sure it's fairly cheap and it's fairly fast. When you get down to this stage, you can do more expensive sorts of things like making a malt, which is about maybe $100 or $200 a sample kind of thing. Obviously, we can't do that on 10,000 samples. So easy things to select for, early in this process are plant height, and so this is a picture of a winter nursery where there's some nice differences in height, and I could select some short ones if I was going after that. We use a lot of genetic markers in my lab, and that's really a big part of my research effort, is to think about how do we use information about the barley genome and genetic markers to make improvements in efficiency and speed in our breeding process. And it's not something I'll go into in much detail, but just to make a note that at a very early stage of the breeding program, we can grow up individual plants in the greenhouse, snip little pieces of leaf, put them in this is a little plastic plate where we put the leaf samples in, and then we can send that off to a lab, a USDA lab in Fargo, North Dakota, that services the Midwest, and they'll extract DNA and they'll run a whole bunch of markers on it, and they'll send us back a spreadsheet with a bunch of data that we can then use to do selection and make predictions about things. So it's a really exciting and fastly evolving technology that we're taking advantage. I'll talk more about winter barley, but that's a pretty easy trait to select for. It's actually one of my favorite traits to select for as a breeder. You just kind of walk out into the field and see what's dead and what's alive. And it's much easier to select for than disease resistance or anything else. So very satisfying, and we can find differences and actually make progress in that, and I'll mention that in a little bit. We do disease resistance screening. This is actually one of our disease nurseries. So this is a nursery just dedicated to Fusarium head blight. And we probably look at 10,000 rows where we inoculate that with those rows with a pathogen and then go out and visually assess them for disease. So it's a very laborious thing, but because it's so important, we tend to do it a little bit earlier in our selection process so we can try to make as much progress as we can. Obviously, we do lots of yield trials, and so we set these up and we do for our first year yield trials, we look at about 300 lines in each of our programs, 2 rows, 6 rows, so on, at 3 locations, and then the second year we'll reduce the number of lines and expand the number of locations, and then eventually whittle that down to a couple of that we're interested in. Malting quality, I kind of mentioned. There's a USDA lab at Madison, Wisconsin that provides that service for all of the public breeding programs, and we can do about 800 samples a year. We have sort of a quota. So we have to make very strategic decisions about which 800 samples we're going to send each year to get that malt analysis done. And they'll malt individual samples that are a few hundred grams, so less than a pound of barley will be malted and goes through kind of a mini brewing process where they can measure some of those important factors for us. And then the last thing is, brewing, which never gets done until it's almost a variety. So a lot of the brewers will tell us, why don't you breed for flavor? And I said, well, we'd love to breed for flavor. If you can take 1,000 of our entries, make beer out of it, taste it, and tell us which ones taste better, we'll breed for flavor all you like. So we have this discussion about flavor, and we have some ideas about how to approach that. But really, the thing that's a real challenge in barley breeding is the end product is really never made until you get to the end of the process, where you have 1 or 2 candidates that you're looking at that meet all the other specifications, and then they go into that. And so we have this testing program, and we do a pilot malting with about 30 pounds of grain. So we're pre-screening, and then when we get to plant scale brewing, which is where it really gets to the brewing, that's where we get 10,000 bushels, and we look at a couple of entries a year, perhaps. Okay, so these are just some, these are just experimental lines that have been in our program. This just gives you an idea of sort of our success rate. And this, I would say, is fairly typical for a breeding program. We put about 4 entries into our AMBA pilot testing, which is the American Malting Barley Association pilot testing each year, and anywhere from 1 to 3 of them might be rated satisfactory in any particular year. And we need to get sort of satisfactory ratings a couple of years to go ahead with it. This line here, M160, looks really good in 2014. It had one sort of minor defect in 2015, so I'm kind of pushing it in there again. It's going to get tested again. I hope that this one is one that we can consider releasing in the next year or so, but it looks pretty good. I'll say a little bit about varietal trials. This information is all available. This is actually this year's information is not quite available yet. We're just putting together our varietal trials. So the Minnesota Ag Experiment Station puts out a varietal trials for all the crops that are being bred at the University of Minnesota. So this is the data that will go into that for barley. We've broken it up into 6-row and 2-row. And what I'm showing you here is the average of the state yield over 3 years, and then these are our main testing locations, sort of multi-location testing locations. And so you can look at just, in this case, yield, and the top yielders are Rasmussen and Innovation. Innovation is a variety from Anheuser-Busch. That's a relatively new one. Rasmussen is one that has consistently been a top yielder, but for whatever reason, the industry hasn't really expressed much interest in it, so it has essentially no acreage, but we still keep it in because that's what we want to beat in terms of yield that's coming through. Quest is one that we released that's, it's not the highest yielding one, but it has some Fusarium head blight resistance, so if you want to hedge against that risk, that's a good choice. And then we have some 2-rows that are currently out there. Probably the one that's really of interest right now is this ND Genesis. It's a new one out of North Dakota's program. Did really well in our yield trials, and has some, and I think the maltsters and brewers are pretty interested in it. So for 2-row, ND Genesis I think is a really good one to look at. Conlin is one that has been around for a while, and it's quite good in terms of having low toxin levels from Fusarium head blight or head scab, so it has appeal there, but it doesn't have the yield potential. These are just some of the other characteristics, and I think I won't really go into a lot of detail. Again, you'll be able to access all this information on the Minnesota Ag Experiment Station website. But we also rank the diseases, and I just want to point out that we rank the diseases from 1 to 9, and we're trying to standardize this across at least the small grain crops, so 1 is good and 9 is bad. And I've sort of color-coded them here in this Excel spreadsheet. So you can see there are some diseases like stem rust where we have really good resistance across the board. There's other diseases like Fusarium head blight or head scab where we've got some work to do. Spot blotch is kind of a mixed bag. The 2-rows tend to be not quite as good for spot blotch. The 6-rows generally are a little bit better. And then this is the bacterial leaf streak I mentioned. Again, there's really not very large differences among the varieties there, and that's something that we're actively working on. All right, so that's snow. Apparently we're going to actually get some eventually this year, but we've had this really interesting long fall. So I want to switch gears and just talk a little bit about winter barley. these are some of the reasons we're interested, and I kind of alluded to them earlier. I think there's potential for increased yield. We could avoid some diseases. There's some potential for weed suppression. This is a picture taken on the same date. This is the spring planted trial, and this was the fall planted trial. So you can see there's a lot of competition for weed potential there. So you can manage those. There's some what we call ecosystem services. So we're trapping carbon when we have things growing in the landscape more. Has some advantages for nutrient cycling, wildlife habitat, reducing erosion. So we think those are some valuable things. And then there's some industry benefits as well. Potential for double cropping, I'll say something about that. Earlier harvest, so you can spread out activities on the farm and that sort of thing. So we think there's some really interesting potential for winter barley in Minnesota, but of course there's a challenge. And so this is what a winter barley trial will look like in a fairly severe winter. And you can see it's quite easy to tell which ones don't make it. And in this particular trial, there weren't very many that had very good survival. So that's our big challenge in winter barley, breeding to increase winter survival. We've done some agronomic studies, and we're continuing to do some with winter barley because we want, by the time we release a variety, we want to be able to give growers, good guidance on how to best be successful with a winter barley. And so one of the studies that we've done is a planting day trial. And so these are 3 varieties that were planted the end of August, middle of September, end of September, and middle of October, so just maybe right around now. And one thing that was kind of surprising to us is that we got better winter survival the later we planted. That was sort of the opposite of what I would have expected. And this has been repeated in several different years. So it looks like there's some advantage to planting later, which from a cropping system point of view would actually be desirable, because we'd like to come in after a soybean harvest and be able to plant winter barley. It looks like on this end of it, this will work pretty nicely. The hard part will be getting the barley harvested soon enough to put in, say, late planted soybeans. But we are actively actually working on that, and I've got a partnership with our soybean breeder where we're trying to think about how do we breed these 2 crops so that they fit a system so we can double crop soybeans and winter barley and keep sort of continual cover on the landscape. And we have some things that we're going to be trialing. We have a double crop trial. We started this, and a relay, double and a relay crop trial we started this fall. So we're going to start with what we have, but we're also going to try to actively breed both soybeans and barley to optimize that system. The other thing that we kind of learned was that we took the same variety and we looked at plots with different levels of survival, and we looked at the yield, and we found that really the yield sort of plateaued out at around 70% survival. So we don't need to get 100% survival to maintain those yields. We'll probably, because barley will tiller if it's a little bit thin, so that makes our job a little bit easier. We can go from 20 or 30% survival up to 70% survival. One thing I wanted to this is probably not well known to most folks. It wasn't even well known to me a few years ago. But our strategy is to breed a facultative winter barley. So winter annuals, true winter annuals, require a cold period in order to flower. So you plant them in the fall, they experience our beautiful cold winters, and that cold temperature for a prolonged period of time tells the plant that it can go into flowering. If you take a winter cereal and you plant it in the spring and it never has that cold period, it doesn't flower. It just grows looks like grass. So that's a challenge for winter breeders, because it means we can only do 1 generation per year in our breeding cycle. In a spring program, we can grow during the summer. I can plant in the greenhouse in the fall, and I can also plant another generation in the greenhouse in January and do 3 generations per year, which is a huge advantage. So what does facultative mean? Well, facultative means that it has winter hardiness, but it doesn't require that vernalization period. And it turns out that vernalization requirement, that cold requirement, isn't really necessary to make it through the winter. What's necessary to make it through the winter is that the plant not start to flower in the fall. And we control that by a gene that is responsive to the photoperiod. So facultative barleys will have, they don't require the cold period, but they do require long days in order to flower. So that way, if you plant them in the fall, they're never going to flower because our days are too short. And then they'll be fine. They'll make it through the winter, hopefully, if we have enough winter hardiness. And then they'll flower. But we can also plant them in the spring, and they'll flower and they'll do just fine as well. And that's also a great advantage from a research point of view, because half of the years we don't get survival in our winter trials, so we get no data. If we can plant them in the spring, we can at least collect some data on disease resistance and yield and that sort of thing. And I would think from a seed producer's point of view, being able to produce seed from a winter annual by growing it in the spring, would be a big advantage too, because you always be able to produce and have an ample supply of seed available. So facultative is really where we focused our effort. So that's kind of a novel approach as well. And I think I talked about that. Okay, so when we started working on winter barley, the first thing I had to decide is, well, we better know what winter is. What is winter? What is it that these plants have to survive? So I'm going to flip through a bunch of these little charts, and I'll describe this first one so you understand what I'm plotting, and then I'll show you a series of these. They're just different years. So what I did is I plotted the minimum temperature for a particular day through the growing season. I think this is like, I can't remember where I started, but obviously this is winter. And then this little white part is the snow cover. So you can see the temperature goes down and the snow comes in. Okay, so this is one particular year. In this year, we use this variety McGregor, which is our fairly good winter hardy variety that's on the shelf that we can use right now. And we had 100% survival in this particular year. Okay, so it's kind of keep that picture in mind. This was 2008, planting in 2008. We had 0 survival, and this was what our temperatures looked like. So a little colder temperatures. We had snow cover, but we had sort of a melting period and then more snow. Here's another year. Again, we had really pretty good survival, pretty deep snow cover for most of the year, even when we had some cold temperatures. 2010, not very good survival. 2011, maybe you remember this winter that never really happened. This was the winter that we didn't really have. We only had, so this pink area is below 0 temperatures. We only had a couple of days below 0 the whole winter. It was pretty amazing. And we had no snow, but we had really good survival, despite the fact that we had no snow cover. 2012, we went back to really poor survival. Again, there was an open period here with some pretty cold temperatures. 2013. So you kind of get the idea. Winters are pretty variable from year to year. Here's 2014, 15. Pretty poor survival in this particular year. Pretty scattered snow cover. And this is sort of plotting them all together. These are the winters where we had pretty good survival, and these are the winters where we had pretty much total winter kill. So in a 9-year history of doing these trials, we got about a 50-50 chance of having decent survival with the current sort of varieties we have. So that's what I tell people right now, to not get their hopes up too high for winter barley. I think we have a lot of potential to improve these odds, both through breeding and through optimizing planting date, looking at no-till so we can trap snow. There's all sorts of other things that we can look at too that will increase these odds. But it seems to have a lot to do with snow cover and cold. So basically, if it doesn't get cold, we're going to be okay. And if it gets cold, but we have enough snow, probably we'll be okay as well. The odd thing is, let's see, I think this was our last winter. So I'll let you guys guess what the survival was this past year, given what we saw there. What do you think? 75. It was actually pretty close to 100. So McGregor was 100%. So again, it wasn't a particularly cold winter. We did get some cold days, but it was, we had a few inches of snow on the ground. So, I think that really tells us the importance of snow cover. If you've got 4 or 5 inches of snow on the ground and it gets really cold, we can probably make it through. So snow cover and temperature are obviously important, but I actually have found the most accurate, and actually it's 100% accurate predictor of whether we get good survival or poor survival, is whether the year that we plant in is odd or even. In the odd years, we've gotten really good survival. In the even years, very poor for the last 9 years. Now, this is 2016, so I just got done planting a huge amount of trials. I'm hoping this trend does not continue because I'd like to get some data out of these trials. But nevertheless, that has been the pattern. But we're trying to come up with a better model than that. I'm pretty sure that's not going to hold up. Okay. And just here's some data to kind of highlight what we've got in our program right now. This was a pretty severe winter, 14-15. McGregor was basically 0. We have some things in our program that are not great, but they're surviving better than our current controls. And these are lines that actually have some malting quality and some disease resistance is what we need. So we're making some progress here. In the mild winter of 2015-16, obviously everything survives really pretty well. We actually can harvest the plots and get some yield data, so some reasonable yields. And then as I mentioned, these are facultative lines. So Maha is a variety that's a facultative. And these two are also in our spring planted trials that are facultative. And so we could see what they yielded, in a spring trial. And it's variable, and we're collecting more data on this. But I'm very curious to see how the yields compare when you plant them in the spring versus when you plant them in the winter and you get survival. And we'll continue to work and study that. Okay, I'll spend a little bit of time on oats. I'm going to skip over some of the history, but I'll just give you a very brief summary of the history of the program. We've had oat research going on at the University of Minnesota, for over 100 years as well. So a long history of that. We had an oat breeder, Dion Stuthman, who was there for quite a long time. When he retired, we did not refill that position, and unfortunately, he even passed away within a year after he retired. And so the oat breeding really kind of stopped about 6 years ago. We continued some work by just getting we did the varietal trials, and we did some basic work in maintaining that program that was shared by a number of folks. But we recently decided to restart that oat breeding program, really stimulated by interest from the company PepsiCo. So PepsiCo owns Quaker Oats, and they've been investing in public research programs in the Midwest because they feel like there's some potential to try to develop, as a long-term strategy, to develop the Midwest as a region where they could source oats, and they want to see, they're really interested in some research in the Midwest. So they're supporting some breeding research and genetics research at Minnesota, but also at South Dakota and Wisconsin. And so we're really kind of reestablishing this collaborative network with the regional states, North Dakota, South Dakota, Minnesota, and Wisconsin, to kind of develop a more regional cooperative breeding program as opposed to these sort of competitive state programs that have been sort of typical of public breeding programs. But anyway, we've got started. I have an undergraduate student who's coming back who's working for us right now, a new graduate student, a postdoc who started, and then I have a technician who's not in the picture. So we've got a team built, a number of different collaborators, and we're going pretty actively into that program. I'll skip over this sort of history stuff here. And just tell you a little bit about what we're doing now. We expanded the yield trials, so we had a few yield trials kind of going in this interim period. We've expanded the number of locations. We pulled out the material that we could find from 2011 when the program stopped, and we planted that out and evaluated it. And so we're looking at that, sort of picking up where we left off. We started making new crosses in that pipeline. We're really working on establishing much stronger and more collaborative regional coordinated effort. We're going to have a new focus on food end uses, and I think that's where our partnership with PepsiCo will really come into play, because they can tell us, just like the malting and brewing industry, they can tell us what sort of things they're looking for in that end use that will make that crop valuable to them, and we can help tailor our breeding efforts to direct things towards that goal. And then crown rust is a huge problem in oats, and so there's work on developing what's called slow rusting resistance to crown rust that we're working on. These are the testing locations that we have for oats. So they're a little bit more concentrated in the south. That one got in here. These are the traits that we're focusing on initially. We don't have quite as many diseases to work on in oat, but crown rust is a really problematic one. This is an example of a pathogen that evolves very rapidly. So what that means is if you develop a new oat variety with a resistance gene to the current races of the pathogen that are out there, in 3 to 5 years, that gene is basically defeated by this evolving pathogen population. So we have this sort of rotating battle of introducing new resistance genes and having them defeated very quickly. And so that creates a real challenge, and that's where the slow rusting comes in. The slow rusting is different from these single genes that we put out in that it's multiple genes, and we're not making the plant immune to the pathogen, it's just the disease develops much more slowly. And the impact that it has is it actually slows the rate at which the pathogen, in theory, would evolve to be virulent on new varieties that are out there. So the idea is that this might be an approach that will give us more durable resistance, which is what we want in oat. We're looking at some quality things. Test weight and grow percentage are probably the two most important things to work on, but then grain protein and beta-glucan are really important for the food industry folks. And then lodging. Lodging is a huge one. So we had 10 trials this year. 4 of those 10 we decided not to harvest because we didn't think the data would be very useful because we had really severe lodging, mostly because of some very specifically timed storms that hit us just at the right time, and so we had things flattened. So we're really focusing an effort on lodging, improving lodging resistance in oat, and this is really a very recent development. I don't know if this will pan out, but we found a person in the engineering department at the University of Minnesota who studies structural engineering, and they use things like CAT scans and things to look at different elements to figure out how they're structurally built. So there's a little bit of research done on corn and using this sort of technology to understand how is a corn stalk structurally arranged. Thinking about it like a building, like building supplies. So we hopefully will get a pilot project where we do some CAT scans of oat stems that are either less or more resistant to lodging, so we can understand how they're built in a way that gets them more resistant to lodging. And if we know that, then we have a tool that we can use in our breeding program to try to select for that sort of thing. So it's kind of a wacky thing that's out there that may not pan out, but it's one of the reasons I really like working at the University of Minnesota, because you have all of these folks with way different interests, and sometimes you'll run into somebody who's a civil engineer who's interested in trying something crazy on oats. So we'll see where that pans out, but regardless, lodging is one that we're really going to work on. We had trials around the state and with our partners also in Wisconsin and South Dakota, so we had lots of data we generated this year, so the program is really kind of up and running, full blast. This is a picture of our rust nursery, just at planting, and the Crown Rust Nursery is a really unique site in basically the world. I'm not going to go into the very complicated biology of the rust pathogen that I can't even remember, but it requires an alternate host, which is buckthorn. And buckthorn is pretty prevalent around Minnesota and other places. So we call this the buckthorn nursery. We have a row of buckthorn planted there, and then we plant a susceptible variety early before we plant anything else. And then in between these rows, we'll plant all of our experimental lines. So then basically the pathogen will move from the buckthorn, it'll infect these things that are early, and then these things will produce spores that will land on our experimental lines. And that allows us to do a really rigorous testing for resistance to crown rust. We're also exploring lots of different phenotyping tools or tools that we can use to measure traits. And a big thing right now in plant breeding is high throughput phenotyping. How can you measure and quantify things really quickly and rapidly? So we're looking at some imaging things so that we can do kernel weights and growths, measuring things like stay green. So I have a student my graduate student is very interested in using drone technology and aerial imaging in our disease nursery. And so this is that buckthorn nursery. And seeing if we can take these images and quantify more carefully the amount of disease in our plots by using aerial imaging and looking at images over time, as opposed to the very laborious sort of walking through the plots and visually assessing each of those plots. So we're pretty excited about using that. We also bought a new plot combine, which will be really great, shared among the small grains program. So Joachim Wirza has one that will do sort of the northern locations, and then we'll be doing the southern locations, and that will allow us to do more plots and get our data faster. I'll finish up with just a little bit of data in the oat varietal trials. And I won't spend a lot of time on this other than to point out that we have a couple of experimental lines that are left over from the Dion Stutzman program that look pretty promising in terms of crown rust resistance. So the data here doesn't look quite as nice as some of our buckthorn data, which is kind of our sentinel area. So a lot of times we'll release things and they'll last for a few years, but if something looks good in that buckthorn nursery, it usually means that it's going to look even better when we get it out on the landscape. So we do have a couple of things in the pipeline that will hopefully be able to be up for release. We're deciding that this year. I think I'll sort of skip over that, but I will mention that, let's see, I thought I had put a URL in here, but there is, if you do want to find any of the information on all of the crops that the U of M works on, you can go to the MEAS website, so it's maes.umn.edu, and they have information on all kinds of crops that you can get listed there. I'll stop there and answer questions if there's time, or...
We got time for a couple questions.
Okay, great. Yeah?
Why don't brewers like grasses and barley?
Yes, good question. Yeah, I was really puzzled by that too, because in addition to its yield, it actually has high malt extract, which is something that the brewers like. They didn't tell me for quite a long time. One person told me, well, it's sort of more discolored than other barleys. But beyond that, they haven't really told me. When Miller was testing it, they actually, when they brewed it, they found a problem with foam stability. So right from the get-go, they weren't very interested in it. Anheuser-Busch said, well, we like it, go ahead, release it, we'll probably use it. And then, like I said, the only thing I've heard from them is an issue with discoloration. So unless you can convince them that you can produce clean, bright Rasmussen grain, I'm not sure, yeah.
Just as a follow-up, is there any relationship between high yield and quality then?
There's not a direct relationship in that sense. Not really between yield, but disease resistance is a problem. So often when we'll make a cross to improve disease resistance, we'll see the quality go down. So there'll be it'll mess up the beta-glucans or something else. And then we have to kind of figure out how to fix that. Yeah. Yeah.
Buckthorn as a host for pathogens, how far away does the buckthorn have to be?
Yeah, that's a good question. The question is how far away, so the buckthorn is this alternate host for the crown rust pathogen, how far away does it have to be? I don't know that I have a good answer for that. I can tell you that in our buckthorn nursery, we get high levels of disease. We have yield trials on the St. Paul campus further away, and we don't see nearly as much disease. So I think it's fairly local. It has to be, certainly, less than a mile probably, or something like that. Yeah, sure.
Thank you.